The overall goal of this method is to provide a microscopy-based protocol for high-resolution imaging and three-dimensional reconstruction of the mouse blood-brain barrier. It combines immunofluorescence of free-floating sections with confocal microscopy to analyze subcellular organelles within the brain endothelial cells.
This method outlines the preparation of brain free-floating sections, immunofluorescence staining, image acquisition, and analysis parameters for high-resolution microscopy of the blood-brain barrier. The main advantage of the technique is that it allows for the quantification of subcellular organelles within cells of the neurovascular unit. Demonstrating the procedure will be Roberto Villaseñor, a Roche postdoctoral fellow from my laboratory.
To begin this procedure, immerse a freshly 2% PFA perfused brain in 20 milliliters of 2% paraformaldehyde for 7 hours at 4 degrees Celsius for post-fixation. Afterward, wash the brain extensively with ice-cold PBS. To embed the fixed brain in agarose, prepare a 3% agarose solution in PBS by heating it in a microwave. Then, gently swirl the solution to cool down but avoid solidification. Dry off excess PBS around the brain before immersing it into the agarose solution in a plastic container. Rotate the brain inside the agarose to remove air bubbles. Upon agarose solidification, carefully remove the agarose block from the plastic container and cut a cube around the brain using a razor blade. Mount the brain onto a vibratome specimen holder using cyanoacrylate glue. After that, transfer the brain in the specimen holder.